So now we will be seeing in details of the promoter region of the eukaryotic system. So promoter region is important for anything, for any gene transcription process. Like we talked about the upstream and downstream, right? So this that is your DNA. We are considering, or we are searching in the DNA genome, like searching for a gene. We're looking into one gene in the genome, and we're looking at the different portion of one gene. So in the transcription, or genes needs to be transcribed. So in the transcription process, we mainly search for, like, promoter is important, right? It mainly it starts the process. So just like then we, after identifying the promoter, then we look upstream or downstream, like how many codes in the downstream, and then we determine how, and then we determine the stop codon, like the UAA or UGG. Then once we get the stop codon, then we can guess that how this long would be the amino acid, and this long would be the nucleotides would be this long. And then in the downstream we search for, from the promoter, the downstream is this 3' end. Going to the 3' end is downstream, this signal, and upstream is by going in the 5' end from the promoter, or from any gene, from any location. If you go 5' end, that will be upstream, and if you go 3' end, that will be downstream. So let's start from the promoter. So core promoter. So promoter has some unique sequences, that is called Tata box, because TIA repeats are present in the promoter region. So that is why, like, whenever we see some TIA repeats, that means we are in the region of a promoter of a one gene. And then the promoter region has another part, that is this one, the transcriptional start site. So this transcriptional start site, so this is not precise. That would be problematic. So transcription, then the promoter site contains transcriptional start site and the starter box. And then we talked about the regulatory regions. If you remember, just upstream is regulatory regions from the upstream of the promoter. So the transcriptional start site from the first nucleotides is called the +1. Then just 25 nucleotides upstream, the approximately 25 nucleotides upstream, the Tata box is found. And then from 50 to 100 nucleotide upstream, this enhancer, this regulatory regions of a gene is found. Regulatory regions is like enhancer or silencer, like those transcription factors, those mainly enhance or silence the transcription, are called the silencer or enhancer or regulatory elements. So we'll just take 1 minute, 1 minute's break. So this is the promoter region. Like, promoter region is we're looking at in detail. So that is why, if we look in the promoter region, for promoter, it is composed of two regions. One is the Tata box, and then there's A and C, adenine and cytosine, and there's five. And in the downstream of A, there is five pyrimidines, that means cytosine CTs. And in the upstream of A, or upstream of C, there's two pyrimidines. So there's no purine. So that is the hallmark, or you can say the general sequence that you can understand the promoter regions. Once you determine or recognize the promoter regions, then you can look for upstream in the enhancer or the regulatory regions, or in the downstream to find out the stop codon for the Like you can easily get find the stop codons, like there's complementarity present, right? So if you get the complementarity in the DNA strand, then you can easily guess the complementarity in the RNA strand. And subsequently, if you determine the codons, then you can determine the sequence of the amino acids and proteins. So that's mean the upstream is going to the 5 prime and the downstream is going to the 3 prime.
So in the upstream portion, why does it call enhancer or silencer?
Um, like we have seen the regulatory regions, like just here, just this is simplified here. If that is promoter, right? In the upstream, there's a regulatory sequence. There's a regulatory sequence, and there are different proteins bind to this regulatory sequence. We focus that we mentioned that this function of the regulatory sequence is to like control or regulate this transcription process, right? The whole process. So how this regulatory sequence controls this process? By like different transcription factors, like different proteins, enhancer or silencer. We are calling those proteins enhancer or silencer depending on their function. Like different proteins or transcription factors, they can enhance or silence. That means they can turn off and turn turn on the gene or the transcription process. So those mainly turn on or the enhance the process are called the enhancers. And those mainly stop or inhibit this process or silence the gene are called um, silencers. And as you can guess, they mainly control this transcription process. So they are called regulatory sequence and they are called regulatory proteins of a gene or a particular gene. And if we can categorize them by the function, like are they increasing the transcription or increasing the gene expression of a particular gene, then they are called enhancers, and the opposite for silencers.
Okay, so thank you.
So, mm, so here just the characteristics of this promoter is mentioned here, written here, but eukaryotic promoter sequence diverse. It can vary. Not all protein coding genes are data boxes. So in majority, in a typical sequence, this data box will be present, but not all the cases. The regulatory elements. So there are also unique characteristics of this regulatory sequence. That means if we can recognize the start site, transcriptional start site, and data box, then we can recognize these promoter regions. And then if we go upstream from 50 to 100 nucleotides, then if we find CG repeats or CAAT repeats, those are called CG box, GC box. Then we can guess those are the mainly transcription binding sites, transcription factor binding sites. Those will affect enhance or silence the transcription of a gene. So that is how we need to identify the different portions of a gene that mainly controls the transcription of that gene. So as I mentioned, the regulatory elements are short chain sequences that affect the ability of the RNA polymerase. That means if transcription factors, if 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 transcription factor, like if that is a silencer, then it will bind here, and subsequently it will recruit other proteins, and this will like those other proteins will bind here. And if proteins are bind here, that means RNA polymerase cannot come here and start the transcription. So regulatory elements affect the ability of RNA polymerase to recognize this promoter sequence and then alters the transcription process. So there are two categories, enhancer and silencers. Enhancers, the DNA sequences recognized by the TFs. TF is transcription factors. Those are proteins that the enhancer mainly stimulate the transcription process, and silencers inhibit transcription. So it is mainly the code promoter for polymerase RNA polymerase 1 and 3 are different. So it is RNA polymerase 2 is shown here. Why 2 is shown here, RNA polymerase 2? Because RNA polymerase 2 mainly involved in transcription of mRNA. So we are focused, we are dealing with mRNA, right? So this is promoter sequence, or this is typical condition for RNA polymerase 2, right? That means the protein coding genes. The RNA polymerase will code for mRNA. So mRNA will code translate into proteins. So we will be more interested in proteins, right? Um, Particularly protein coding sequence of our genome. So just remember that RNA polymerase 1 and 3, RNA polymerase 1 will be involved in ribosomal RNA synthesis, and RNA polymerase 3 will be involved in transfer RNA synthesis. So here it is shown for RNA polymerase 2, which mainly encodes for transcribe the mRNA. Now, just we have to think outside of the box, that we know this is the RNA, right? mRNA. So in the mRNA, this is the start codon. That means that in here the translation process will start. That means the start codon will. That means AUG OG. Like if you play PUBG game, the OG gun. If you remember, those OG gun or AUG mainly the start codon here. So the start codon will, like like if you can determine the AUG, then you can start codon. You can recognize the start codon, and you can recognize the RNA, protein synthesis, like first amino acid. And then if you go downstream, and if you find a stop codon like UAA or UGA, then you can stop there, and that will be your last. Like stop codon are not translated. Start codon is translated, but stop codon is not translated. So now the question is, if a mutation changed the start codon to stop codon, would this mutation affect the length of the transcribed RNA? So that means within your RNA, like not RNA, like within your genome, like within your DNA, some mutation has occurred, and those mutation, whenever the mutated gene is transcribed, this will converts this start codon into stop codon. Then what will happen to this transcribed RNA? The length. Will the length be affected? Just think about RNA. So if the start codon is like replaced or somehow mutated into stop codon, so the length of the RNA, will that change? RNA length will not be affected, but like if you consider the length of protein, what will happen? So two situations here. One for the RNA, like mutation is occurring at the DNA level of DNA, and then that gene is transcribed into RNA. And as a result of the mutation earlier, in the transcribed RNA, the the start codon is replaced, like the start codon is converted into stop codon. Then what will happen in the translation process? Here, here is we are saying that the start codon is replaced by stop codon. That means the start codon, that means there's no start codon. So that whenever the ribosomes come, just like no one has told, the ribosome ribosome will be there, but when the uh, transfer RNA will come, it will detect the stop codon. So there will be no adding of the amino acids. So that means there will be no protein. So shorter version will not be there. That means there will be no protein of that gene. So that's mainly some of the uh, some of the rRNA mainly tries to like microRNA, they mainly inhibit the protein synthesis, but not this way. Just like they just try to like this is mRNA. So the microRNA is the function of the microRNA is to inhibit this mRNA, right? So the microRNA somehow bind to this mRNA and form a duplex so that they cannot enter in the ribosomes cannot detect. So this is other mechanism, but we are saying here the somehow the mutation in the abnormal condition has occurred. So here, if the start codon is replaced by stop codon, then there will be no protein synthesis of the gene.